The severe acute respiratory syndrome coronavirus 2 consists of 4 main structural proteins, including the spike, nucleocapsid, membrane, and envelope proteins. To date, most research on SARS-CoV-2 has focused on the effects and changes present in these proteins, as they are essential for the survival and pathogenesis of SARS-CoV-2 infection and are highly visible, thus making them excellent drug or vaccine targets. However, several other SARS-CoV-2 proteins play a role in the survival of this virus during infection. To this end, researchers in a recent bioRxiv study discussed the effects of the SARS-CoV-2 nonstructural protein 1, which binds to the ribosomal subunit, thus inhibiting translation. Study, parsing the role of NSP1 in SARS-CoV-2 infection. Image neighbors. Study findings. Previous studies have shown that SARS-CoV-2 NSP1 can inhibit translation and promote ribonucleic acid degradation, whereas residues R124 and K125 are required for the inhibition of translation. To further assess whether NSP1 is also necessary for messenger RNA degradation, 2 NSP1 mutants were generated. For the generation of NSP1 delta RB, amino acids 155 to 165 were removed, whereas the development of NSP1-CD required both R124 and K125 to be mutated to alanine. NSP1 delta RB failed to be associated with ribosomes, while NSP1-CD could associate, but much less effectively than wild type. Interestingly, the level of NSP1 was lower for weight than the 2 mutants, thus indicating that it inhibits itself. When the expression of green fluorescence protein reporters either fuse to host beta-globin 5 UTR or the 5-leader sequence of SARS-CoV-2, the weight and NSP1-CD could both inhibit GFP expression in the host, but not in the leader sequence. To further examine the effect of NSP1 on translation mediated by an internal ribosome entry site, a bisystronic mRNA that allows expression of 2 luciferases to be mediated by either cap-dependent translation or downstream encephalomyocarditis virus internal ribosome entry site was used. As expected, the weight NSP1 was the most effective at suppressing both luciferases, with NSP1-CD showing a lesser effect and NSP1 delta RB showing no effect. To characterize the cellular transcripts sensitive to this degradation, the CoV-2-5 leader sequences upstream of the NSP1 constructs were fused to prevent self-inhibition, which showed that weight with the CoV-2 leader was refractory to its self-inhibitory effects and that the mutated construct increased expression. SLAM-seq, which is an approach that allows for the measurement of endogenous mRNA, was then used to measure the induction of mRNA decay by NSP1. This assay revealed U to C mutation rates at 1.5% that subsequently rose to 5.5%. Cells expressing weight NSP1 also showed a more rapid turnover of RNA as both time and the portion of labeled RNA increased. Cells expressing NSP1 delta RB or NSP1-CD showed no reduction in cellular mRNA half-lives. A SARS-CoV-2 mutant was generated with the same amino acids deleted. Vero and Calu-3 cells were then infected with both weight SARS-CoV-2 and the mutant. After confirming the expression of NSP1 was not altered, the propagation of the virus was examined. Both cell types could support both viruses, with similar viral titers in Vero cells, but much lower titers for the mutant in Calu-3 cells. This finding suggests that NSP1 helps SARS-CoV-2 survive in the immunologically active Calu-3 cells and indicates that NSP1 interference could affect the interferon response. To confirm this, a JAK-STAT inhibitor was used to prevent IFN signaling, which successfully rescued CoV-2 MUT propagation in Calu-3 cells. To further investigate the role of NSP1 in SARS-CoV-2 pathogenesis, a Syrian hamster model was used. 2 groups of 8 hamsters were infected with either weight SARS-CoV-2 or CoV-2 MUT and were weighed daily, with viral lung titers quantified after day 7. COVID-2 MUT-infected mice exhibited less weight loss that became statistically significant at day 6. In addition to reduced weight loss, COVID-2 infected mice also exhibited significantly lower levels of viral lung titers that were below the limit of detection at 7 days post-infection. Effects of NSP-1 on translation and accumulation of viral and host mRNAs. A. Protein synthesis measurement by flow cytometry of Calu-3 cells infected with COVID-2 weight, or with COVID-2 MUT for 4, 5 and 7 HPI, or an uninfected control following opropargyl puromycin incorporation, and fluorescent labeling using click chemistry. B. Cumulative frequency of human and viral genes according to their relative translation efficiency in cells infected with COVID-2 weight or with COVID-2 MUT at 4 HPI. T was calculated from ribosome profiling and mRNA sequencing, and is defined as the ratio of ribosome footprints to mRNA for a given gene. Each dot represents one of 9 major viral mRNA species. Conclusions. The authors have successfully shown that NSP-1 inhibits both CAPT and IRES-driven translation. Furthermore, the researchers here identified the residues that are essential to these processes and demonstrated that NSP-1 is vulnerable to inhibiting itself, which can be prevented to increase expression. Further analysis demonstrated that NSP-1 RNA degradation is dependent on ribosome binding. Investigations in Vero and Calu-3 cells proved that NSP-1 is important for viral survival, that the mRNA degradation effects remain active during infection, that the loss of this protein in vivo alters the pathogenicity of SARS-CoV-2 infection. Taken together, the findings from this study help identify NSP-1 is a key protein for the survival and effectiveness of SARS-CoV-2. In the future, this information could assist researchers in developing novel therapeutics to reduce the severity or length of the disease in coronavirus disease 2019 patients. * Important notice. bioRxiv publishes preliminary scientific reports that are not peer-reviewed and, therefore, should not be regarded as conclusive, guide clinical practice, health-related behavior, or treated as established information.